We're going to get started, so if people could take their seats. Again, I'm Sharon Terry from Genetic Alliance. I'm the chair of the workshop here on behalf of the Planning Committee, which I think I have to say I'm very, very pleased with. All of our hard work over the last few months really has generated an excellent workshop, at least as far as we can see so far, and I think today will prove to continue that and, in fact, add more substance to the kinds of conversations and discussions we had yesterday. I'm not going to attempt to summarize yesterday's conversation. It ranged far and wide for those of you who weren't able to be with us. We did hit a lot of really critical pieces. We did do what we asked, which is to really be truthful and honest, to bring out the elephants in the room, to find the places that we did need some work as well as those in which we had some agreement. We will hear from the chairs of all of the sessions later this morning, and they will give us a 5-minute summary of each of those sessions, which will be a yeoman's task even in just each of the sessions individually. A couple things I would just call out. I think we heard widespread agreement that data sharing is critical. We heard some question about whether we've sort of just been practicing using comparator arms and placebo arms, and that maybe we need to look more critically at the issues that are surrounding using sharing all data and what is considered the pre-competitive space. We also heard attention, I think. There were some people who said, it's easy, it's just the culture, and there were other people who said, it's very hard and very expensive. So I don't think either of those is wrong. I think they're probably both right, and I think there's probably a lot of nuance there. It struck me yesterday that there really isn't a kind of dichotomy where one end is this and one end is that. There really are a lot of areas of gray in all of this that we're going to need to sort through. And the final remark I'd make before I'm going to turn it over to Bob is I think we really do want to keep in mind why we're here and what we're here for, and that the patients that we keep referring to as them is us, and us is them. So I think even if we can consider that in our language today and think about when we do say we need to communicate with them, that's us. We are all, at the end of the day, patients, people, citizens, individuals who care about health, and that while we do have professional hats, and mine happens to be always the public, the citizen, the patient. I think we all want to keep in mind all of the stakeholders as we work through these things, and to consider, and today we will, some of the more cutting edge kinds of ways that the public is engaged. This is a field that in fact has not had the same kind of consumer involvement that other fields or industries have had, and as that involvement becomes more and more commonplace, more powerful, more integrated, I think we're going to see some sea changes that are going to be pretty abrupt and pretty dramatic. And I think the work we do here can lay some great groundwork for that to move on. That said, I'm going to introduce very briefly, the same as yesterday. We are not going to do extensive, the biographies of everybody are in your packets. We're going to do very brief introductions. So I'll introduce the next chair, which is for session 4, Incentivizing Policy and Cultural Shifts to Enhance Data Sharing. So today we're going to see some of the policy, governance, funding, those kinds of issues really highlighted. And so Dr. Robert Harrington will chair this session. He is the Arthur L. Bloomfield Professor of Medicine and chairman of the Department of Medicine at Stanford University. Thanks, Bob.
Thank you, and good morning. So I'm just going to make a few remarks to open up today's session, and first to do that, to reflect upon a little bit of what we did yesterday. And as I looked through the sessions and thought about them, what we did yesterday, we really had 3 broad sweeping concepts that we covered. We talked in the morning a lot about the benefits to sharing data, using words like liberating data. We spent a fair bit of time with a technical approach to different data sharing models and went through some examples of how different groups might collaborate and share data. Then the afternoon was largely devoted to the much more technical aspects of data standards and some of the governance around how we achieve data standards. But it was interesting throughout even the most technical discussions yesterday that the phrase that, well, it has to be done within the culture of that community kept coming up. And so that led in perfectly to today, where we are going to largely focus on culture And we have two broad goals today. We're going to explore some policy issues, and we're going to see some examples of policy from outside the U.S., from the journals, from academia. And we're going to have our speakers address some of the cultural expectations that exist in their world, be they academia, the government, the funding bodies, etc. There'll be case examples from all of these groups. There'll be some plea from patients as to what their roles might be in this culture of research. And one of the things that, unlike yesterday, where, again, a lot of technical jargon was used, today the words that we're going to use are policy, practices, attitudes, facilitation, collaboration, and a key word that there'll be a whole session within this session, incentives. How do we get people to do this? I love the comment yesterday when somebody said, well, talk nice to people, ask them friendly. Say please, thank them, include them. And I think you'll hear a lot of those messages of incentives throughout the course of the session. So I'm looking forward to this session. As I said, it's a lot less technical and a lot more about how do we actually get down and convince each other that sharing data amongst ourselves is a positive thing. So first, we're going to hear from our colleagues across the pond. Hans Eichler is a senior medical officer in the European Medicines Agency in London, UK, and he will be talking about some of the initiatives of the European Medicine Agency in trying to achieve clinical trial data transparency.
Thank you. Good morning. Thanks for inviting me. Just by way of introduction, what is the European Medicines Agency? For those of you who are not familiar, just think of the FDA, CDER, and CBER. We're in the business of regulating drugs and biologicals. The analogy is not perfect, but broadly it's sufficient for today. So, I'll try to give us our perspective and a bit of history, recent interesting history we went through. I don't know whether you followed that in the press, probably not on this side of the Atlantic, but we had a bit of an issue with transparency. And we had some unflattering remarks in the press, and it was not a good time. And at the end, it ended with this one here. Has been given a rap over the knuckles by the European Ombudsman. What is the European Ombudsman? The truth is, I don't know exactly what that is. It is an institution that is like a court. It isn't a court, but it's like a court. And you can go there if you have a complaint, and the Ombudsman will then say, you go this way or that way. So we had our experience with this. It's an office. It's also a gentleman, but it's an office. So then we had a lot of press from the not so favorable press again, from the academic community. Maybe some of you have seen this in the British Medical Journal. It details the travails that a group of academics had with us. We dragged our feet. Anyway, it was a long process, and maybe Peter will talk more about that, I don't know. But the end of this was about a 2-year process. The end was pretty transformative. And it's in this sentence. We're a public body. As a public body, like here, we are obliged to be fully transparent. We have to give everything away. If you ask me about a document, you should receive it. Exceptions are if it is personal protected data, that hasn't changed, or if it is commercial confidential information. And the big sea change was this sentence. That's what the Ombudsman said. There is an overriding public health interest, therefore, clinical trial data is no longer considered commercial confidential information. I'm fully aware of the huge implications this has. So, I hear our chairman today, and I saw it on the program, that you were talking about incentives for data sharing. Well, we're beyond that debate. We don't need incentives. That is our legal basis, so fine. We're incentivized. That's behind us. But the next frontier is coming. So, let's go back again and look at this evolution of transparency. And you know all the top bullets up there. The third bullet is access to documents. That's retroactive availability of trial reports. So as of last year, if you asked us, please send us all the clinical trial data of drug X, we would do it. It is a pain in the neck because we would have to redact it to see whether there is personal, not commercial, personal data in there, but you would get it in paper format. So it's there, but it's not very helpful. The next step is we will proactively publish trial reports. There are people who say, you're putting out these, we call them EPARs, European Public Assessment Reports. So when we license a drug, there is an assessment report, like from the FDA. Why don't you put a hyperlink in there electronically where I can click straight on the full trial report? Fine, there's nothing wrong with that. Technically, it's a bit of a challenge and we're not great with IT, but in principle, we could do it. And the last bullet is, of course, this one. Public availability of raw data. So, say, give us all the line listings, patient-level data. Yes, probably you will get it. I will say, well, not just probably, we will make it available. And the thing is, when we talk about data, it is all the data we own. Not own, but we hold. So that is pre-licensing RCTs, but there will also be pharmacovigilance, observational data, the whole set. So that's where we are going, gingerly. And that puts a number of people outside their comfort zone, because it's playing with data. And you all know that data is like children. You like your own best, and you don't like strangers to play with them. Understandable. So, an industry is particularly, shall we say, outside their comfort zone. It reminds me of a book that I read, and this is a whole new paradigm. And I recommend the book. It's about reinventing discovery. It came out last year. And, excuse me, they're talking about the division of labor, how historically, thanks for that, historically, observation and analysis have been yoked together. You are a researcher, you do the experiment, you analyze it, you report it, end of story. And the exceptions already, to some degree, have been regulated products like medicines, because there you had a second party to reanalyze, that's the regulator. But now we are moving into a completely new age, a new division of labor. If everything is out, everybody can reanalyze, play with it, do whatever they wish to do. So is that a boon or is it a bane for drug development and public health? I will start with the negative so that I can end on a positive note. So don't think I'm just negative. The cons. Of course, there are data protection issues. And the interesting thing is, in our history, recent history, we heard, as I said, from this European ombudsman, you have to be fully transparent, give it all out. At the same time, we have another European institution, a public institution, that is something called the Data Protection Officer or something. It's another office. And they say, wait. There is personal data protection. So these two public bodies will then have to fight it out how exactly we're going to do this. But that probably can be addressed. I don't think this will be a major issue. What we probably will do, I say probably because I cannot predict the future, but it's most likely. We will tell the industry, look, you have to be aware as of date X that all of your clinical trial data that you submit to us will be available to anybody else. Therefore, ensure that there is no identifiable patient data in there. That's probably the way we have to go. But then there is something, so that's probably an addressable problem. But then there is that second one. Phantom risks, health scares, and we will be blindsided. We don't like that, of course, industry as well. So what we're going to hear or see is a paper in the Lancet Monday morning saying that this drug or this vaccine causes the side effect. Regulator, what are you doing? And our response will be, well, we haven't read the paper. Where is it? So an unpleasant situation. Is that a real risk? I think it is. And I like this paper. I always quote it from my colleagues at FDA who said, well, we have entered an era of increasingly frequent publication of meta-analyses that all show some safety signal, true or false, and there is always this call, regulator, do something. So what we're going to see is many more third-party publications that will drive the agenda. Many of the drugs we licensed will come under attack for true or false reasons. I give you an example of what can go wrong. And I use an example from the US background because this is a US audience. There was a drug that showed to be beneficial. It's tiotropium. It's still on the market. And then there was a meta-analysis in 2008 saying that the relative risk of cardiovascular events is 1.6. And that's classic. You always get these relative risks between 1 and 2. And then what do you do with it? It's observational data or meta-analysis data. So there you are. And that is a real red flag. And that is a risk to a drug that is potentially beneficial. Luckily, in that case, that company had underway a study that looked at the long-term endpoints, the clinical endpoints. And that showed that there was no risk. If you look at the relative risk, it's all below 1. So if anything, it might actually be beneficial. So, but if that study had not been done, I don't know where that drug would be. What will we do in that case? We'll see such cases. And here is another quote that comes from this institution. I like it very much. That was recently published. There will be challenges to achieving or to doing RCTs once there is this red flag of a risk. Why would patients want to enroll? So we will jeopardize perhaps useful treatments that we have. Is that in the interest of public health? No, certainly not. I think it is a real risk. But, so that was the negatives. The positives. What we're going to is open science, basically. Anyone can have any of the data. And that will be a boon for a lot of cutting-edge technologies that are only around the corner that we haven't much experience with. There are predictive models for patient selection to appropriate treatments. You need data-rich sets to substratify patients, to find what treatment would be most appropriate for patients with this set of characteristics versus the others. There are a few studies who've done that, but I see a lot of potential in there. Machine learning systems, you take these large data sets to derive new information, again on risk stratification, benefit stratification. Ultimately, you could even think you could match a patient history to a clinical trial data set. So that would be the greatest. If you have a patient, you punch into a computer all the patient variables, and you say, for that patient, most likely on that huge data, based on that huge data set, this is the right therapy. And then there is a last thing. I know comparative effectiveness research is high on everybody's agenda. We call it relative efficacy assessment, same thing. And there is the siren songs that we will have head-to-head comparisons. I don't believe in that. It's just too expensive. I don't think it's worthwhile. What we need to is probably indirect comparison assessments. So something along the line of this. We by chance had 3 anticoagulants authorized almost at the same time, both in the US and in Europe. And these people did indirect comparisons. They did not have access to the full data set. They did it based on the aggregate data of clinical trial reports. And if you talk to them and say that is a clear weakness. Have we had the line listings, we could have done probably a much better job. So I think this will be a boon for comparative effectiveness research. So, where are we going? That is now EMA again, European Medicines Agency. We have announced that we will have a workshop on the 22nd of November this year, so in about a month and a half, where at the EMA, there will be 200 people or so. It was open to the public, but now we're oversubscribed, so you can no longer apply for a seat. Where we will invite pretty much everybody, all our stakeholders, industry, Cochrane collaborations, critics, public citizen advocates, whoever. And there will be a podium discussion, and it will be a free-for-all. And we will just listen. We will not talk too much, because we will have to develop our own policy, how we make those data available. And we want to establish a working relationship with those stakeholders who are willing to engage. So what are we likely going to do? And again, I say likely because I cannot predict the future. Look at the first statement here. While protecting the decision-making process, that is polite code for saying, okay, you will get the data once we have assessed it. Once we have reached a decision, then we make it available. What we don't want is analysis come up while we're still in the assessment process. And we see papers, I don't know, in the equivalent of the New York Times or whatever, saying that they should never authorize this because it's dangerous, they should, or whatever. That is not helpful to fulfill our role. So once the decision has been made, the drug has been licensed or rejected, then we will let it out. And what we will have to develop jointly, probably with our stakeholders, is the standards for storing and sharing of data. I think you just said it's very difficult technically. I think it will be. It's not trivial, but it will be doable. So the technical part will be addressable. We will have to debate the level of data to be released. And I know, again, I was asked not to talk about raw data, but that's where we are now. Nobody knows really at what level that should go out. Should it be individual MR readings or CT readings? All open. We have to debate the standards for protection of personal data. That's a no-brainer, of course. We will maybe have to discuss, that's more critical already, the quality standards. If someone wants to do meta-analysis, comparative efficacy assessments based on these indirect comparisons, What are quality standards? Should there be a voluntary standard by which everybody should abide? And then the last one. That is probably the most critical. Rules of engagement. You could say, yes, you can have it. All the data are out there. But it's not just you take it. You have to give something before you take it. So it could be open but filtered. What we want, basically, is a pre-registration of protocols. What we want to avoid by that is data dredging. So I get all the data on one drug. I let my computer program run overnight. I program it in such a way to stop when it has any finding at the p less 0.05 level. I wake up in the morning, I have my breakfast, and I write my paper. That's not what we want to see. So tell us what you're going to look at. Are you going to do an exploratory study or a confirmatory study? Deposit the protocol, and then you have it. But all of that will need to be hammered out. So we are in that process. If you want to watch our meeting on the 22nd of November, it will be webcast. There is limited spaces, I think, but it will also be recorded. So you will, in December, you can have it and watch it on our website. Okay, thanks for that.
Thank you very much. A good start to the morning, and we'll have plenty of time for questions at the end of this session with the panels. Next up is Andrew Vickers, who we heard a bit from yesterday from Sloan Kettering, who will be talking to us on clinical research data sharing practices and attitudes. Andrew?
Yes. Where's the clickers here? Yeah, so I don't know if there are any junior faculty in the room, but if you want to get invited to the Institute of Medicine, perhaps it's the paper in the New England Journal of Medicine that you don't need, or the Journal of the National Cancer Institute, but something in the New York Times. This is something that I wrote out of frustration. There's a lot of consumer advocates here. I guess I'm a consumer because I'm a statistician, and there's this thing like you like your own data best. You don't want other people playing with it. Well, that's what I do. Is I play with lots of kids. I play with data, and whether it's my data or other people's data, that's not what is important. And so I wrote this paper. I don't actually know that much about data sharing. They were talking about HL7 and CDISC yesterday. I had no idea. I just come from it as a statistician trying to do research. I really believe that the more people we have looking at data sets, the better off we'll be. And I was getting very frustrated. This was a good few years ago, but 2008, over the first few years of the 2000s, I've got an extra few minutes now.
Right? I took it away from you.
There you go. I'm Andrew Vickers, and So what was the sort of typical experience I was having? So somebody said, could you design a study for us? Could you do a power calculation? I'm like, oh yes, I'll need the variance of the endpoint in the placebo group. There was this trial published at the NIA. You know what? I could call those guys. They could send me the data set. I could get the variance, could do the power calculation. As an NIH researcher, NIH-funded trial, I am not prepared to release the data from the control group at this point. That was it. Blunt rejection. This was another experience I had. It's a big problem when you do meta-analysis, when you combine data. So we had a whole bunch of trials that said, did the patient experience, basically did the patient throw up during chemotherapy? The frank emesis is actually the term that they use. And then this one trial, very nice trial, I think it was published in JAMA even, said, well, how many times did they throw up, the mean and standard deviation? We couldn't combine the two bits of data, so I wrote to the PI, who was great, and she said, it's a great idea to do a meta-analysis, it's very important. I'd love to send you the data, but my statistician won't allow it. This was a Cochrane review. I think it was actually published eventually in the Journal of the Clinical Oncology. How is that helping science? I don't understand this. We're trying to put all the data together, right? You never learn, the baseball season just ended, right? So meta-analysis is when the baseball season ends, and you can't look at individual games. You've got to look at the big picture. Anyway, someone talked about, Dr. Eichler, is that how you say it? Dr. Eichler was just talking about looking at data sets to build predictive models. So this is something that myself and a colleague, Mike Catan, were looking at in the very early 2000s. And we were trying to come up with some ideas of how you would mine existing randomized control trial data to say who would benefit and who wouldn't. And it's all very technical statistical theory here. And we needed a chemotherapy data set. And so I remember a 1-hour phone call with the PI of one of the NIH-funded, federal money cooperative groups, and they were just, "Whoa, we don't know about this," So we started begging, right? This is roughly what we said. We'll do anything to get this data. We will corrupt it. We'll give you veto power. We'll corrupt it. We'll expressly state in the statistics in case a patient goes and reads in Biometrics, this technical journal of statistics. We'll say, look, you can't draw clinical conclusions we never heard back from them. We weren't able to get this data. So look, when I wrote all this stuff, I wrote a paper in Trials, and I wrote this in the New York Times. I was treated by some statisticians as the kid that no one would play with in the schoolyard. He just is unpopular. But I want to say I do this all the time, right? I mean, I think probably I'm half successful, half unsuccessful, but I'm PI of several very, very large collaborations that have been very, very successful, have led to some very, very interesting results. The bottom one is that really we got a lot of institutions to provide their data to MSK. I think it was a total of 9 different institutions to show that there are absolutely massive differences in cancer recurrence rates between surgeons depending on their level of experience. It's actually in Europe. It's actually starting to change practice in the way that surgeons are trained and the way that they organize specialty care. So I've also reanalyzed, individual randomized trials. This was from something called the Prostate Cancer Prevention Trial, a large NIH-sponsored trial. This was from a Scandinavian group. These are all interesting findings. This current guidelines, or for years, would say that how your PSA, which is used to detect cancer, how that changes, its velocities, can tell you whether you've got cancer. We reanalyzed this data, said it doesn't. It's come out of guidelines. Here we showed that actually overall most men with prostate cancer don't need to be treated. But if you do need to be treated, yes, you really need surgery. And this was very, it's what everyone had suspected. It was confirmed when we reanalyzed the largest randomized trial that had been done to date of surgery versus observation. So what are the arguments against data sharing? Remember, I'm mainly dealing with academic data. I very rarely approach drug companies for data. So what do the academics say? And I know not just because of anecdotal experience, but with Caroline Savage, who's now Caroline Bennett, we published this paper where what was the experience of trying to actually get data, all from academics. Published, and we chose it in PLOS because Public Library of Science says, you must give the data. If you publish with us, you must get the data. And we actually found only 1 in 10. We approached 10 individuals, 10 papers. We only got 1 data set from a journal that said, you must share your data. So we know what everyone said, right? So what were the sorts of things that they were saying? I'd love to help you. I really support what you're doing, but it's very busy. I'm just teaching now. I've got a grant due and so on and so forth. Wait a minute, putting together a data set. You just published a paper. What was that based on? You've got to do this anyway, right? I'm a statistician. I publish papers the whole time. We have a clean and well annotated data set. And if anyone wants that data set, I'm like, here it is. We've sometimes had that. We published a very controversial paper comparing surgery with radiation oncology, and the radiation oncologists were freaking out. Well, here's the data set. Have a look. It's totally understandable. Just to give you an example of one of the things we did, here was a study we published in one of the BMC journals, and you can add additional files, and you can download the file, and we put it in Excel format. I just cut and pasted it from Stata, which is a statistical package, into Excel. And then the variable descriptions, so that you could actually understand it, were also cut and pasted from the statistical software, because I've got to have this too. I've got to write this up for myself and for my staff. So that was about 5 minutes to actually, it wasn't $2.3 million or whatever they were telling us yesterday. We've actually done this, that learning curve papers, the 15,000 patients I was talking about, we actually, as an experiment, we said, well, here's a very complicated data set. There were 6 or 7 papers published on the basis of that data set. What would it take for us to de-identify those data, put them in a format anyone can understand, and publish all the code we used to analyze those data. And that is, we have a lot of tricks like subroutines that make everything look nice, but it's kind of difficult for somebody else to follow, so we had to take out all those subroutines and rewrite them. And you can find this paper. We reckon it took us about 8 hours of work, right, for 7 major papers in the academic sphere. Okay. Second argument against data sharing. I'd love to help you, but what about patient confidentiality? It actually reminded me once of my wife. She rang up, she got an appointment with the doctor, and she's just called on the phone to the secretary and says, oh, can I just ask, does he have, board certification in whatever it was? And she was told, I can't tell you due to HIPAA regulations, right? Which just means, like, can I have some coffee? I can't give you the coffee because of HIPAA regulations. It's just like whatever. It's like a defense against anything. Okay, so just in case anyone's aware of this, let's go through step by step. Here's a data set. On the left are 3 columns that in theory could be used to de-identify them. If you select them by selecting column A, B, and C, and hit the delete button and then the save button, we have now de-identified the data set. I do accept that it is sometimes more complex. This is the complete code we had to write to de-identify a data set. It's things like, I don't know if you can see that, does it have a laser pointer? I don't know. Yes, here we go. Young men and old men. Because prostatectomy is rare in young men and old men, if you were very young or very old, we reset the ages. If you were 32, we made it 40. If you were 78, we put it 75, okay? I'm not stupid. I understand HIPAA. This is all the code. I mean, how much code is this to write? How long do you think this takes to write this code? It's really not very complicated. I mean, probably took less time than one of those conflict of interest declarations that you have to fax around the place. Another argument against data sharing, other researchers may conduct invalid analysis. Oh my God, something invalid happening in science. So this is personal interest to me. I recently published a paper. It was a meta-analysis. These guys, where did they come up with this idea of finding out how to do publication bias, right? There was a correction for publication bias, how many unpublished studies. They just pulled it out of their nether regions, okay? Why did we do this? Because it was in the study protocol that we'd published. We explicitly, 5 years ago, said how we were going to correct for publication bias, okay? Now, why was this person able to say RCTs of 100 subjects? It was because in the paper we said only if there are 47 unpublished trials with 100 subjects would there be no difference between groups. Okay, so the person was only able to criticize us because we actually told us what they did. So of course we just should do this, right? We looked at the problem. We don't think it's a problem. If we don't say what we did in the paper, then no one can criticize us. And it's exactly the same with data, right? No one says don't publish your methods or results because people might do invalid critiques, right? Same thing in data. Publish your data, let people critique you, and then you can have a sort of scientific debate about it. Someone might so this is more of a sort of drug company thing, right? Now, I haven't really dealt with too many drug companies, but I have heard arguments about it, in the New York Times article, we point out that there's someone called John Kerwin, who is a rheumatologist from New Zealand, who's actually written to drug companies and researchers and said, would you share your data? And that's very nice to come here and have people from Sanofi and the drug industry say, yes, we'll share our data. Because back in the early 2000s, 75% of drug companies and researchers said you shouldn't let data be open. And this is one of the reasons. Someone might delve through our data and find out our drug didn't work. Well, how's the secrecy working out for you, right? What about this? We looked at our data, and then, we really looked very hard to see if there were side effects, and then we made it available for everyone else to look at, okay? And we really couldn't find something. Something happened. We're sorry, we took all reasonable steps. What are you going to do? You cannot sue someone if they take reasonable precautions against something. I think data availability, I'm not a lawyer, but it sounds to me to be a pretty good defense against litigation. Okay, secrecy has not helped the pharma industry, either in terms of just pure finances, I think, what did Merck cost? Like, it cost them billions. I mean, billions and billions of dollars, something like $5 billion in litigation and fines and so on, or in terms of their reputation. Fifth one, data sets contain proprietary information. If anyone can explain this to me, maybe can you send me an email or put your hand? I don't understand what the proprietary information could be, really, okay? Maybe there's some marker in the data and you show the drug affects the marker and so you want to then build it, God knows, right? But I mean, this will be rare. If it happens, well, you know, oops. You know, if it is true, then maybe someone could make an argument that there is a small piece of proprietary information and we do want to hold it back. We're going to give you the rest of the data, okay? Researchers have a right to exploit data they've spent years collecting. I agree with this. This is an argument against data sharing. Okay, we do need to incentivize people to collect their own data, but you can get over it, right? So we published the learning curve data set. I will always be the learning curve guy, right? I published the first paper. I don't really have to worry that my academic career is going to be derailed because someone does some reanalysis of something. It's very easy to have some kind of embargo period, publish your trial or whatever it is. You've got a year or two to exploit the data and then anyone else can have it. And of course, academic credit. I mean, if someone were to publish a paper on the learning curve, you know, I'd look terrific, right? And in fact, I think there's some people talking about how you can get academic for credit for reused data. We do know now that papers that publish their data are cited much more than papers that don't. So there's already an incentive there. So this was a paper we published, this is a good few years ago, in Trials. All these codes of conduct, what should you do with data? I mean, they're not difficult to work out. Things like, if you're going to publish a new analysis, you've got to contact the trialist beforehand, okay? Or they should be, you know, journals need to offer a commentary and, you know, so on and so forth. I think these are all solvable problems. So get used to it is my message, right? I mean, I was born in 1967, which was, I think, Loving versus State of Virginia. At the time, most Americans thought interracial marriage was like, come on. How could you even think about this sort of thing, right? It's exactly the same. Open access to papers, when that came out in the late 1990s, early 2000s, there was outrage, right? Clinical trial registration, okay? That was seen as a whole big issue that would damage the commercial interests of companies. A whole bunch of things seemed like very radical things at the time. I think data sharing is just one of those. Thank you very much.
Thanks, Andrew. If people weren't awake before, I suspect they are now. So we're going to turn now to some of the policy issues, and Steve Goodman, the Associate Dean for Clinical Trial Research at Stanford School of Medicine, is going to talk from the perspective of research funders and publishers.
Hi. So yes, I'll be talking about them. I'll also be talking about academics, and you'll see there might be some question as to whether Andrew and I coordinated. But we didn't. We're just on the same train traveling somewhere. Let's see, how do we do this? Okay, there we go. So actually, I'm going to be talking about not just about journals and funders, and I'm going to be talking about this thing called reproducible research. And I haven't heard that word used today. That is the dominant word that's being used in the biostatistical and bioinformatic community, and I'll talk about why. Also, you'll notice I changed my affiliation here from Stanford to the two journals that I work at, Annals of Internal Medicine and Clinical Trials, and why is that? Well, first of all, just telling you what some of my experiences, but in fact, journals themselves represent, there's a conflict of interest for those that are not what's called open access. I actually don't like the term. I say open access for who? I say that the journals who will accept without charge a submission from anybody, that's open access too for the contributors. So it's just a question of who pays and how the money flow goes. But for subscription-based journals, you don't get free, not everybody gets free access to their content. They hold it back because they have an interest in it. And it serves another interest. I mean, it enables them to have editorial staffs and add value, and I believe it has added value. And that's the same kind of disincentive that that Andrew was referring to when he talked about the incentive we need for people to produce data in the first place. If they're going to spend 10 years running a trial or whatever, they, if they're just going to hand it over to somebody else who's going to produce their papers in 6 months, that's a huge disincentive. So these are real things, and they result in value, but they also result in a disvalue. And how to balance those, I think, is one of the real challenges. Now, there actually was a conference here on this almost exact topic 9 years ago. I participated in that. I wasn't on the panel. I talked there. And they endorsed something called the upside principles. And I'm not going to read through these, but it could be an exact mirror of what we've been talking about today. These upsides stand for Uniform Principle of Sharing Integral Data and Materials Expeditiously. I don't know which they came up with first. But it's really a wonderful document, and it includes a lot of excellent suggestions for how data should be shared from the perspective of the publishing community and the scientists who generate it. And these upside principles are formal principles that have been endorsed or not by a variety of other groups. So I would encourage you strongly to look at this document because it presages a lot of the things we talked about today, although there's been a huge amount of cultural and technical change since then. So let's talk about some of the purposes of data sharing, and I'm going to focus on a purpose that actually hasn't been the main focus here. First, it's to discover new things, and that's been almost the dominant conversation. But the second is to assure that the study was correctly analyzed and interpreted. That is, to provide assurance that the chain of scientific custody from protocol to conduct to data to analysis to published result is sound. And I've often said, sort of, if we could see under the hood of what's going on in the analysis of a lot of studies published in major journals and minor journals, we would just blow the top off of science. It's unbelievable what a mess it is. It's a huge mess. Only the major journals have sort of teams of methodologists that can even begin to check this, but it's hugely challenging. And the journals that go right below that tier, really very little. I mean, they depend on peer reviewers. Peer reviewers are totally powerless. And why? So here's an example why. This is a study called the CAMP Study, Childhood Asthma Management Program study that was published in New England Journal. And these slides are courtesy of Jim Tanasha, who is a PI on this study. And there's nothing wrong with the study, by the way. I'll tell you that ahead of time. I'm just going to show you what went into the study. And here we go. The number of separate forms they used for the study, 72. Number of form revisions, 109. Number of data entry forms, 41. Total forms in the database, about 300,000. These are the number of transactions in the database, 35,000 changes, 4,000 deletes, about 330 total transactions, and it said almost 300,000 total forms. In terms of the actual manuscript, originally it started with 73 tables and 9 figures. There were 40 revisions. What was published? 3 tables and 2 figures. And this was published in October 2000. Now, how do we begin? How do we begin from this tiny little slice that we see to begin to work backwards and figure out, well, is what they did right? And it's this sort of thing that I think that folks were talking about when they talk about the complexity of sharing what was really done in many studies, even though I completely concede that for many studies I've done, they're as simple as Andrew described. So what was the result? Well, the result is this comment that was actually made by a computer scientist who started the field of reproducible research. He said most published papers are an ad for the research rather than a real report on the research itself. That's absolutely true. Now, with supplementary materials and appendices, this is changing, but we still can't get to the heart of it. We just can't get to the heart of it. So there was a paper that inspired our thinking at Annals about how to deal with this, and there's a lot more to the story than I'm going to have time to tell you today. But this was a story on this topic. Do Oscar winners live longer? Now, you might wonder, why does the Annals of Internal Medicine, why are they interested in this? Now, most of the patients of our readership are not in the running. But it was published. I would say we were a little uncomfortable with it. It claimed that there was a 4-year survival advantage, and any of you who watched the Oscars have probably heard this claim every single year. It's trotted out, and you'll hear it again this year. And I have to say we were pretty skeptical about it, and there's, again, a long story. But we actually asked the author if he would publish the data set publicly upon publication of this, because we wondered about it and we want to know what was under the hood. He did for one day. It appeared on the day of publication, then disappeared, he said, for reasons of confidentiality. I don't know if the Oscar winners are so worried about confidentiality, but in any case they did. But this is what happened. This is how he explained the possible 4-year advantage. Personal chefs, trainers, nannies support ideals of lifestyle. Constant scrutiny and entourage invested in the movie star's success leads stars to, quote, avoiding disgraceful behaviors and maintaining exemplary conduct. Well, I actually had to remove the few slides I had showing the exemplary conduct of some movie stars because I didn't think it was fit for this august institution. But let's just say that doesn't always occur. But anyway, there was another statistician, Jim Hamley, who was a little exercised about this because he didn't believe it and he wanted to get the data set. He tried. He was not successful for 5 years. He finally got an amended version of the data set because the author said that they had lost the original one or couldn't find the original one. So he got an updated version, and he was convinced it was misanalyzed. And in fact, I would say that it was. And he published a very detailed explanation of why and how to analyze data like this. And he came up with this result, an estimated life expectancy difference of 0.7 years with a p of 0.16. And thereafter, I and the editor published a little mea culpa about how it happened. But he says, of course, readers and commentators should be doubly cautious when they encounter statistical results that seem too extreme to be true. And we published this because we thought it was an important corrective. First of all, it was a very important methodologic point about how to do this. It had to do with immortal time bias. But it taught us a lesson, and we think about this a lot. And there was another paper that was actually published the same year that gave us the tools. It was this paper called Reproducible Epidemiologic Research, and this was by 3 colleagues of mine when I was at Johns Hopkins, where I was for 20 years until last year. And they talk about the research pipeline. Again, this was derivative from prior work in this area of reproducible research. And this is sort of bringing it into the life sciences realm. It really was in the world of computer sciences and computational sciences, where the models were so complicated that even if you had the model, it was very difficult to figure out if you had it right. So just having the model, some of these economic models, I mean, you can't figure out from looking at them if what they did was reasonable. I mean, and that's a situation where you have everything laid out in front of you. So here's the pathway, and actually you'll see this is from biostatisticians. At least it starts with the measured data. Of course, we're interested in a lot of things that come before the measured data, but the article is at the end. But we want to shine a light at least on this middle part, the analytic data and the computational results, because it's not a given that will produce the results that you see. And so part of what you want to do is that. But the other issue that they brought up here is sharing models. And right now, the models of sharing is you share or you don't share. And they introduced the idea that, authors put stuff on the web, supplementary materials, or some central database, and the readers just download the data and figure it out and get the software and run it. That's the current model. Well, they propose 2 other ways to do sharing. One is that there are all shades of gray between the share and the don't share. I think this is extraordinarily important. The dialogue so far has been a little bit impoverished because, and I think John Wilbanks and Devin McGraw coming after me may address these issues, that there are ways of sharing, and we do it all the time when we use intellectual property of many kinds, that don't involve complete free use of what you get. And so here are models. The data can be used for purposes only of reproducing the results. Share alike. It can be used to produce New findings or results, but any modifications to data, including transformations, etc., have to be made available under the same terms. Or maybe you can't publish without the co-authorship of the people who provide it. Attribution, data can be used for any purposes with authors cited. Full access can be used for any purpose. And these are contractually mediated. So I'll look forward to the discussions after this. But and I'm sure there are other shades of gray. The point is there are ways to mediate this relationship that are not, I give you the data, I don't give you the data. And maybe we're going to have to start working on that. The other thing they talk about in that paper, which I'm not going to be showing you today, is the new form of producing reports through software called S-Weave, which are dynamic reports, and it's incredible to see. They're not. It's not just results on paper the way we see it now. The code is woven into the paper, and the data sits behind it. And for example, if you change a single point of the data, it ripples through the entire document. All the figures change, the analysis change, the tables change. So this is what we actually have right now. And so everything is transparent. You have the data, you have the computations built into the paper. So that's another way of addressing this issue about the issue of just downloading the data and sharing it. The code and the analysis is already built into what you get, because those actions are very difficult. There's also a lot of different data sharing structures, and this was not in that paper, but I just want to point out we talk about, we don't make these distinctions. There's of course the dyadic, the researcher and the person who wants to use it. There's the institutional way of handling it, where we have central repositories, and then we have the study-based ways of handling it, like MESA or Framingham or the Nurses' Health Study, and then of course we have this new model that's being developed of the nonprofit institutional open access public data sharing. And we've heard Dryad mentioned. We haven't heard iDash. Dryad, as I understand it, it was built for the life earth sciences, but Dryad, iDash, supported by I think it was ARC or NIH, is specifically for health research, and they have developed not only automatic anonymization. You'll see up there integrated data for analysis, anonymization, and sharing. But and this is going to be very, very critical to a proposal I'll put out at the end. An ID, a unique ID for every data set, and this is actually the technical development I think that we're going to have to solve this problem. So iDash is at UCSD. It's developed by health researchers, and they have a long list of health data sets that they already have online, and there's some really exciting stuff going on there. So we finally did publish this paper in 2006, Reproducible Research, Moving Towards Research the Public Can Really Trust, and we proposed the following thing, that there's be a reproducible research statement at the end. Now, we talked a lot about what we wanted to require. In the end, we really could require nothing. The only thing we could require was a statement, is whether the study protocol will be available and how, whether the code will be available, and whether the data set will be available and how to get it. A couple things about that. Well, why did we just say, this is about as weak as you can get? Now, why? And this is a key to the issue of what the solution is. It's because if Annals of Internal Medicine starts putting up big barriers saying you must do what you don't want to do, well, what's the quickest thing you're going to do? You publish somewhere else. So, journals are competitive with each other. They also want to publish the best stuff. And they can't put up barriers that nobody else is putting up. So we felt that this would at least shine a light on this and make the statement public. But, like warning labels on cigarettes, people get used to this, and they gradually are not too embarrassed by saying things will not be available. And that's a big problem. One thing about the protocols, to tell a little anecdote, one of my favorite is a group that we, couldn't figure out what they were doing, couldn't figure out. So finally, we asked for the protocol. We waited for the paper. Like 5 months later, we write to them, say, what's going on? Where's the protocol? He said, oh, don't worry, we're still writing it. So this is a reproducible research statement, and this was brought here's another one, and this represents one of the problems you'll see. and this is very, very typical, study protocol and statistical code available from Dr. McWilliams. Now, what you don't know is what happens when you actually write to the person. I'll talk about that in a second. But what they've said is, what about the data set? They direct you to N. Haynes. N. Haynes, and that's often what we get. So they don't give you the analytic data set they use for this particular study, and this does not help you very much. We did a look back. This is actually done a few years ago at 2 years of this, and about over 80% said the protocol was available, 70% said the code, and 60% said the data were available. And we're actually in the process of the editors and staff there of doing a much longer follow-up. This is like from 2008 or 2009. We also looked what happened. We emailed the authors and asked them, were people asking for these things? And, about, maybe 5 to 10% said that they were getting some requests. We do that with 100% of our authors now, but we're trying to think about how we can really find out what the experience of requesting these things are. But the uptake is not huge. I mean, for the most part, they tell me their impression from the canvassing is that not a lot of folks are getting the requests. Now, this is a very comprehensive list of what's going on at all the journals, and I'll just highlight a few. And up at the top, I have, do they require that the code be shared publicly? By the way, on this issue of code, one of the issues of, blowing the top off science, there are a non-trivial number of contributors who don't know what code is. I mean, they point and click, they use Excel, who knows what they use. But when you ask for statistical code, they literally don't know what you're talking about. So this is, again, a problem and shows how even asking the question can be revelatory. So New England, background for this. So one of the first things that the Annals did after they instituted this, they went to they're part of the ICMJE, the International Committee of Medical Journal Editors, and brought this up and said, is this something we can all agree on? And without going into further detail, the answer was no. And it was voted down that year, and it was taken off the agenda for discussion this year. And so it's not a subject of discussion there at the moment, and journals are basically deciding on their own what to do. So New England Journal, as far as we could tell, they have no requirements, although I was just told that they actually are now, for the past year, always posting protocols, and I'll show you that in a second. JAMA, nothing, except they do require the somewhat controversial an independent academic statistician to corroborate analyses of industry-funded studies. This does not mean, by the way, that they might not occasionally publish protocols or whatever in the supplementary materials, but we could find absolutely nothing on the website or in articles that showed. Now, they led the way in issues of how to report author contributions and things like that. So it's not that they're doing nothing in this area, but they've approached it in other ways. The Annals and BMJ actually picked up exactly our model, and they require the author to state whether these things are available. The Lancet, we couldn't find a policy identified. Biostatistics, the journal, has a very interesting thing, which is they have a full-time reproducibility research editor who's actually the first author, Roger Pang, on that epidemiology piece, who will actually rerun the data if submitted that way, and they will mark with a big mark on the paper, D if the data was provided, C if the code was provided, and R if it was proven to be reproducible. Exciting idea, but it takes a dedicated resources and a dedicated editor to do this. No small thing, and not something that we can expect journals to be doing in general. PLOS Medicine has a very comprehensive policy. I don't have time to read all through this, but one wonders exactly how it's working in practice. But it is by far the most comprehensive written policy that we have. This is an example of the kite mark up here. This is the code, and it's very, very prominent. And something like that. We thought of doing that at Annals, but didn't quite think of this. It was like, one thumb up for one thing, 2, 3, and then we thought, well, maybe asterisk. Okay, no, teddy bears, whatever. We couldn't get away from the idea that the authors will feel that they were being judged, and it would look bad. So we couldn't do this. And interesting, they use a sort of categorical rating system. It's not A, B, C. It's just DCR indicating what folks are providing. So I went to the New England Journal website to see how this protocol thing was working because I could find nothing in instructions for authors about the protocol. So this is last night. This is what you'll see if you go to the website today. And so here's a nice clinical trial. And so I went to the supplementary materials, and there it was. It was just in supplementary materials. It didn't say protocol, but when you clicked on that, you saw this. The trial protocol has been provided by the authors to give into additional information about their work, and here it is. Oops. Dr. Eichler, I don't know. Is this something you could help us with? So what did I go to? I went to Google Translate, and this is what I get. So you can read through that. The previously durchgeführten. Okay, so you know how these things work in practice and how they work on paper are sometimes two very different things. You know, the letter of the law, they absolutely adhere to it. Now, what's also interesting is that in the hard sciences and in basic science, this is a well-established approach, and there's much more of a culture and regulations about data sharing and putting genome sequences and genomic data into databases, and they've reaped a huge amount of benefit from that. But it's a different culture, different regulations, and I would submit a different set of considerations. But we have a lot to learn from that because anyway, so nature, science, and PNAS all have quite detailed descriptions and requirements for data deposition. So I'm not going to go through this. This is the proposal from Science, which basically says, you know, put data into the public repository, you know, and all the different ways to handle this, the code, etc. So what should journals do and what can they do? Well, we've already seen an example of what they can do. Clinical trial registration. This is, you know, a collective statement by all the journals. Here are the editors from all the journals, and we know how that's worked. Well, why has that worked so well, even though it's certainly in need of improvement? Why was this successful? Well, there was a central repository, only one place. You know, the proposals that the journals house some of this stuff, it doesn't work because what you're going to get is different protocols, different data sets, you know, all sorts of different things in different places. That's even if they had the technical capacity to do it. That's you have to develop some sort of curation or whatever. It's a mess. So this is not going to work on a journal by journal basis. And the vast majority of journals are run out of the hip pockets of societies or individuals. They do not have the resources to do this. So we need a central repository. It's very helpful to have a legislative mandate for actually doing what you're doing. And of course, with clinicaltrials.gov, it's not for everything, but it's for an important subset. And this got the ball rolling. And it was used, it's used by many folks for which it's not absolutely required. Really, it was the journals that put the final, I don't want to say, the final nail in the, well, what's the right metaphor? Yeah. And this was a form of journal collective action, which we are not remotely getting in the data sharing realm. And of course, it also required Deb Zarin. So it requires people, structures, and laws. What can they not do? They can't be effective acting alone. They can't be the custodians of the research data. They can't afford staff just for this purpose, and they can't be the sole guarantors of scientific quality. What are the funder policies? I'm actually going to leave this to the speaker behind me. But, of course, we have the Shelby Amendment, which says you can FOIA any research used for public policy. NIH says you have to have a data sharing plan for high-ticket research, but whether this is being honored in the breach, I suspect it is. I don't think we're getting a huge amount of yield from this, and whatever they're getting, I don't know how much it's being used, and we'll maybe hear about that. Interesting, NHLBI requires, and it's not quite the same as the NIH, the deposit into BioLink 2 years after publication, 3 years after the end of follow-up. Again, I don't know exactly what the status of that is. ARC and VA, we couldn't find any specific plans, and NSF just started that you're required to have data sharing plans. Interesting, Howard Hughes has a very detailed data sharing requirement. And it adds these other requirements that as a function of sharing, you can insist on collaboration, that you can require the recipients of the materials to pay for the cost, should include all the data, and provide the executable code and file and source code. Interestingly, all the funders on this list got together earlier this year to try to tackle this problem. And they published a statement, I don't know how many of you read it, that was published in Lancet. And what did the statement say? Here's what it looks like. It was published in January. And I'm not going to read through it, but I'll just summarize it, which is just say, this is a really hard problem and we're all going to work to solve it. So, it's an important first step, but they acknowledge what's important when you read through this. They acknowledge the real interests on both sides, that this isn't just a cultural behavioral problem. It is a community scientific incentive problem that we're going to actually have to work hard to solve. And here is one proposal to solve it. Oh, so how do you solve collective action problems? I don't know how many of you have seen this. These are guys looking at mice dressed a lot like us, and mice are not known necessarily for their cooperation, and this is what they're saying. Okay, now let's slowly lower in the grant money. That's how you get that's how you get collective action. You align the incentives. So here would be one incentive, and it's a sort of slightly more it's the operational version of what Andrew just said. Currently, there are just two ways to use publications to measure academic success. Either are you on the paper, or is the paper cited? That's pretty much it. And there have been lots and lots of metrics sort of built on this, the H factor, the impact factor for journals. Journals themselves are measured. What we need is something measurable, and it's unbelievable what happens when you start to measure things. We need a third measure, which is a marker for the use of the data, for original research published by others. And we can't do this unless we actually develop some sort of unique identifier for each data set. So just like a PubMed ID for the paper, we need the same ID for the data set. And every single time that data set is used, it needs to be put in the paper that uses it, and you can easily go and retrieve the use of the data. And just imagine if you had not just the citations to the original paper, but every time it appeared, because those papers are diffuse. So every time it was used, even by a meta-analyst, that gets reported, and then that goes on the CV, the academic researcher. Now, this is actually being worked on by the folks at IDASH, so this is not a completely new idea, and I think there are other folks as well. But I think it's absolutely key to solving this problem. Just hammering people over the head and saying you're behaving wrong, you're believing wrong, you're acting wrong is not going to do it when there's a whole host of incentives and that go the other way, which is very much also, they would say, about scientific quality and scientific yield, because they know their data best. They construct large teams that are built on the idea that these can be mined, and there's a whole academic superstructure built on the idea that if you put in a lot of work, you're going to be able to reap the benefits of that, and society. And it's not just themselves individually for many years. So we have to create a culture and a reality where people benefit as much from everyone sharing their data for all purposes as they currently do from protecting it. So with that, I will stop, and I apologize for being over time.
Thank you. We're going to do a quick chair switch here, or have our next group of speakers come up. We're going to use you guys in the panel, so maybe the additional speakers can add on at the end. And in the next section, before we get into our discussion, we are going to have three presenters who are going to talk about incentives for data sharing. So really building upon some of the things that Dr. Goodman started with. And our first presenter will be Peter Dashi from Johns Hopkins School of Medicine. And Peter will be talking from the academic perspective of some of the incentives to share data.
Thank you very much for the invitation to be at what I think is one of the most important meetings we're having in the last few years on this topic. In terms of cultures, what I'm going to be describing, I hope, will give you a sense of the gulf between the worlds of academia on one hand and industry and regulators on the other, through the lens of their understanding about different types of data, different types of documents. So I want to focus on the question of what does it take to credibly evaluate trials. Let's start with a typical question. Does drug X reduce the chances of horrible outcome Y? Now I could look at case reports, I could look at case series, I could look at observational studies even, but I decide that since there are randomized controlled studies that address this particular question, that's where I want to look. And so I'm searching across the ocean here for balls. And each ball represents a clinical trial. The red balls are negative trials. Drug didn't work. Green balls, positive trials. Drug worked. Now I could look only at the green balls, could pool them together, but that would be a little biased. So I decided that I want to look at all the balls to be as unbiased as possible. Now how many of you think that's a good idea? Look at all the balls. Minority? No, majority. Okay, good. But it turns out that there's more balls than appear at first sight. Many trials are unpublished, below the waterline and largely out of sight. This is the problem that we know when we call publication bias. Luckily, we've made pretty good progress in this area with registration initiatives like clinicaltrials.gov, although that's still limited to newer trials. But we can now at least identify some balls that are under the surface. However, addressing publication bias retains a fundamental assumption that obtaining a trial, a publication, is the goal. We in academia for years have been calling publications trials. The publication is the trial. This is what clinical practice guideline writers, systematic reviewers, WHO, CDC, and many others have been doing, going around and collecting journal publications for all the trials they could find. But what has over the last few years become a matter of growing concern is the fact that the publication is not the trial. Each trial, as everybody in this room now knows very well, and we've discussed, contains a myriad of documents and types of data. Things like case report forms, things like individual participant data, IPD, internal documents like marketing assessments, investigators' brochures, emails, meeting minutes, and the clinical study report, a CSR, which itself contains many elements like the protocol, the statistical analysis plan, sample case report forms, and so on. Now, all along, people likely had some sense that such documents existed. But how many of us have actually seen these and actually read these? Could I have a show of hands of the people who've actually read a clinical study report? 15, 20? Here's my question. Are any of these documents, or perhaps all of them, needed to credibly assess trials? If all we have is a journal paper, this means that we're putting our entire trust that everything we need to know about the clinical trial is accurately reflected and fits in the 10-page document up at top. That little guy above the waterline. Now this illustration, unfortunately, could only fit so much. So here's a longer list of all the kinds of documents, mostly produced by a trial sponsor, that exist, particularly for industry-sponsored trials. And so I'm going to lead you through some of these documents, showing examples, and continually asking this question: do we need this type of data to credibly assess a trial? Also keep in the back of your mind that data are not just numbers. Sure, measurements are a key part of data, but data can mean analyses, and they can also mean narratives. Both written narratives, as in what happened to a particular patient, but also correspondence with trialists. My argument is that we need all three, and in great detail, to fully understand a trial. So let's start off with a clinical study report, and thanks Dr. Eichler for this report. This is for one trial of Tamiflu, and it's 8,545 pages, and don't worry, I printed double-sided. Here's the corresponding publication. 7 pages. Now, I'm sure you're all thinking that there's no way on earth anybody would read 8,545 pages. Is that what you meant when you raised your hand? No. Okay. So you'd be right. If we look at how a clinical study report is actually laid out, and this is the table of contents from one, we find that it's a highly structured and well-organized document. The first 233 pages of this particular one tells us basically what you would find in a journal article, but it's a big journal article, 233 pages. Background, methods, results, discussion. That's what got compressed to 7, maybe. The next 190 pages gives us supporting trial documents such as the protocol, blank case report form, statistical analysis plan, and certificates of analysis. The remainder 8,000 pages at the bottom is mostly an archival print copy of individual participant data. Protocols show us how the trial was to be run and how data was to be collected. They provide far more detail than a methods section in a journal article. This is from another example from the New England Journal website where the protocol is posted, journal article, methods in the article, 2 pages, protocol 121 pages. Protocols are so long, in fact, that they need their own table of contents. Protocols can also include pre-specified statistical tests in a section called the statistical analysis plan. And they may be in both the narrative form as well as in including statistical code that will be used to analyze the trial results. Access to study protocols help reduce the possibility of reporting biases. It's an original study protocol that allowed investigators of the class study published in JAMA to know that the trial in JAMA had been misreported, and it misreported the original protocol, statistical analysis plan, the trial duration, and conclusions, including the key claim that Celebrex was safer than alternative treatments. Protocol amendments are just as important as the original protocol. In the protocol I just showed you a minute ago from the New England Journal website, it turns out that the authors had only posted version 6 of the protocol. Included in version 6 is a compressed list of changes, amendments that were made to prior versions. The amendments show that one of the pre-specified efficacy outcomes was altered 3 years into the duration of the trial. This is not mentioned in the journal publication. Clinical study report may also contain certificates of analysis, which are an authenticated document certifying the constituents, quality, and purity of the products that are being tested. In this example, we have a record of the appearance of Tamiflu and placebo capsules. The Tamiflu capsule, as you can see in what's blown up here, versus the placebo capsule. The Tamiflu capsule had a light yellow cap, but the placebo had an ivory-colored cap. These details are neither present in the journal publication, here, nor are they present in the body text of the clinical study report, both of which call the placebo matching. Now, we've talked much about individual patient data, IPD, up to this point in the conference. And IPD is commonly believed to allow for the verification and reproduction of trial results, as well as enabling additional analyses. But there is a basic assumption here that IPD is trustworthy. Please recall that it is the case report forms created before the trial begins, and a blank copy of which are included in clinical study reports on which the actual patient data are collected. And sometimes the IPD computerized database does not reflect what is recorded in the case report forms. So this example I have here comes from an FDA medical officer who reviewed the Avandia record trial. And he found that although the patient, you can see here, is recorded to have had a myocardial infarction, that was never referred for adjudication, and so simply disappeared from the so-called raw data, that is the IPD. To credibly assess trials, we might need to look beyond what people typically consider data and into more narrative forms of documentation. How about marketing assessments? Swaroop Vejla, who I see in the back, has shown that before embarking on trials, Park Davis conducted what they call marketing assessments to determine whether the company would aim for producing a journal article, what they call a publication strategy at the end of the road, or a submission to a regulator for an additional indication what they called the indication strategy. In this case, Park-Davis chose the publication strategy and decided they would publish the trial if the results were positive. Well, why might this matter for a doctor or patient analyzing this trial? Well, if it's known at the outset of a trial that no regulator will be scrutinizing the data, there's less of an incentive to make sure the trial is conducted to the highest possible standards. I'm not saying this occurred in this particular case, only that there's less of an incentive by virtue of the fact that the trial appears to have occurred with the assumption that nobody else would be able to scrutinize the data. What about correspondence? Joe Ross, who's also here, and colleagues, looked at the internal correspondence to the STEPS trial of Neurontin. They encountered a letter from the clinical research organization conducting the study, written to Park-Davis, informing the sponsor of numerous problems with the data. The CRO noted to them that investigators are inexperienced with conducting the trials, that they do not have study coordinators, and that training for completing the case report forms was minimal. The lesson here is that it's not measurements taken within the trial that tip us off to these problems, but a letter written about the trial, which allows us to understand that data quality may be severely deficient in this trial. CSRs, yesterday we had a comment about CSRs, that CSRs may be a good first low-hanging fruit in terms of data sharing. And I think this is an important and a good suggestion for the initial step. In fact, there's a really good reason for doing this. They already exist, they're already complete for almost all of these trials. And this sentiment was echoed recently in JAMA in an editorial by Rodwin and Abramson. They noted, again, that drug manufacturers already produce these reports. And importantly, that making these CSRs publicly available, and I would annotate here making full clinical study reports publicly available, would not be expensive, yet disclosure would promote research integrity, medical knowledge, and public health. In a way, though, it really shouldn't be a plea or a dream about what we can do in the future. The experiment has already begun, as I hope you got from the first talk this morning. Since late 2010, the European Medicines Agency revised their access to data policy. It's actually access to documents, any documents they hold, policy, and they've released, it's reported, 1.5 million pages of clinical trial data. For our particular Cochrane review, we received 25,000 pages, including that 8,000 page report I showed you. I'd like to know, put out there for discussion, FDA's position on non-disclosure today when the same data, the same drugs, the same clinical study reports are being released in Europe. But EMA does not have all the data, such as case report forms for all patients, nor do they have all the drugs that we're swallowing. And so I end with the question that I've been repeating all along. What is or are data? And what types do we need to credibly assess a trial? Are we going to settle for polished 10-page journal articles? Or do we need to start thinking about the real universe of trial evidence? Thank you.
Thank you, Peter. Thank you, Peter. Next, we're going to hear from Beth Kozel, who's an instructor of pediatrics at St. Louis Children's in Washington University School of Medicine. She's going to talk a bit about the challenges in a rare disease area, with both the challenges and the incentives and disincentives for the entire community in that regard to want to share their data. Thanks, Beth.
Thank you. Hello, my name is Beth Kozel. I am a pediatrician. I am a clinical geneticist. I am a researcher, and on most days I like to believe that I'm an advocate for the families and patients that I take care of. As was said, I don't have anything else to disclose. I participate in research in a rare disease population. So the group and the families that I work with are individuals that have a syndrome called Williams syndrome. Williams syndrome is a rare disease that occurs in approximately 1 in 10,000 individuals. It is caused, in this situation, it is a rare disease where the genetic mechanism of the disease is known. It is caused by the deletion of 26 to 28 genes on the 7th human chromosome. And the characteristics of the individuals with this condition are well described. So they include characteristic facial features, as you'll see from the cute kiddos and adults here. Significant cardiovascular anomalies, including stenosis of great vessels, hypertension, and as we're learning, also stiff vessels. There's a characteristic neurocognitive profile with very decorated speech, very social activity, but with high anxiety, high and poor ability to have in-depth social interactions. Interestingly, the duplication in this area leads to speech delay and many autistic-type features. There is also failure to gain weight in infancy, consequently leading to this being a presentation often for these individuals in childhood and why they are diagnosed. However, these individuals then go on to have a predisposition for obesity and diabetes, and there are other endocrine abnormalities, including hypercalcemia, thyroid dysfunction. This constellation of features leads to a complicated health picture for these individuals. It also leads to the coming together of many different research groups and research interests that have interests in many of these different developmental, neurocognitive, cardiovascular, all of these different phenotypes. As I said, the molecular mechanism for how this disease is caused is known. However, there's significant variability in the disease that exists. So my particular work is in the cardiovascular effects of this condition. And what we know is that around 30% of individuals with Williams syndrome will have severe enough vascular disease requiring surgery in order to keep them alive. And sometimes even surgery isn't helpful in that regard. And 20% of the individuals will go and live their whole life with none of the vascular problems that are seen, even though the deletion in this area is basically the same deletion. Consequently, there are likely other effects, genetic and otherwise, that contribute to this variability. Now, as a clinical geneticist, I know that when we make a diagnosis of Williams syndrome, we sit down with a family and we give them a list like this of the things that might go wrong. And you can imagine, as a parent, that hearing a list 200 miles long of things that might happen to your child and not really having a roadmap as to where to go from there is a big challenge. The goal would be to be able to use genomic information or environmental information to be able to give people individual information about their child. What are the things that this family, this child is going to have trouble with, so that families can focus their interest, get to the right doctors, and being taken care of in the right way. The problem in doing some of those genomic studies in a rare disease is that the number of samples required to do these analyses are huge. So this is a statistical calculations put together to look at how many individuals are needed to look for an odds ratio of a particular effect based on a particular polymorphism. So what you can see are these numbers here. So if you have an odds ratio of 2.5, in order to get about 80% power to look at that, you need 250 to 300 individuals who have the phenotype and 250 to 300 of them who don't. Now, as you can imagine, if you have a relatively common condition, I could look in this room, I could stand on the corner, I could probably find that many people with hypertension while standing there in the course of an afternoon. I could probably stand on that corner, however, for the rest of my career and never obtain a number of people with Williams syndrome to meet these calculations. So what this leads, and what I have found as a relatively junior investigator going to meetings and listening to talks, is that people then collect multiple, their small group of individuals, and they attempt to do a study with the 50 to 100 people that they can find. They attempt to publish it, and they are met with, you don't have enough power to do this study, so they bow their heads, go back to the lab, and that's where the data sits. Then another group has the same great idea to do this study. They look at their 50 to 100 people, are not allowed to publish it, and go back to their labs. And I've seen on multiple occasions the same projects being done again and again, sometimes leading to negative results, which are then done again and again, and that data never gets out there to benefit families or even to benefit additional investigators from not doing the same study again and again. So this then brings into the role of other groups and how they may work to help facilitate collaborations between investigators, and how that can be helpful and sometimes not as helpful as we might hope. So as I said, in the work that I do, I work with the Williams Syndrome Association, and this is a family group that brings together families and other individuals who love people with Williams syndrome. It has information for families on how to take care of their kids, and information for teachers and people who want to work with them, but then also includes a registry that allows families who are interested in research to get involved and to directly interact with researchers. So the Williams Syndrome Association understands their role in facilitating research and decided to go to their researchers to try and help this collaboration of bringing together researchers that are interested, researchers that are doing these projects, to see if we can't do a better job of collaborating our work together. So this is an example of the work that of a letter that they put out to around 30 investigators known based on their publication records and their participation in family meetings where researchers come to interact with individuals with Williams syndrome, asking them what types of specimens they collected. So in their eyes, this was sort of a first pass at seeing what's out there, what do we even have? So the letter went out to around 30 individuals, like I said, who were known based on their publication records to have samples. Of those, 15 of them were returned, so we're busy. Of the 15 that were returned, 9 of the people said, we have no samples. 6 of the individuals said, we have samples, but there are significant barriers to sharing. Of those 6 people, they each said that they had around 10 to 100 samples, and they did go on to cite some of the reasons that they felt were challenges to sharing those samples. But there were absolutely 0 investigators who said, yes, I have samples, and I would love to share them with you. So, barriers to clinical data sharing. Some of them we've talked about already, and most of all of the individuals actually who cited the trouble with sharing the samples they had talked about discussions with problems regarding consent and IRB. Now, like I said, these are rare diseases. Some of the investigators that are involved in these studies have been collecting samples for 30, 40 years. Some of these individuals were consented before the molecular diagnosis was even known. Certainly, there was no exome sequencing. Certainly, there were not the technologies available. Going back and getting these people to reconsent them and find them based on new technologies and new studies would be a significant challenge. In addition, there are questions of samples conducted years ago or collected years ago in someone who is a child and is now an adult. They obviously might want to have an opinion on how their samples are used. And there are questions of taking this genomic information, putting it in a database, both from a genetics perspective and if you're giving phenotype data, especially when you have a small, tight community where individuals do know each other. Are there potentials for identification? Other things that we have talked about a little bit are sort of closer to home in terms of academia, how we are getting credit for these sorts of things. If it is going to require all of us to put our samples together in order to get one statistically significant paper together, how are we going to get credit? How am I, as a junior investigator, going to get tenure based on my participation with my few samples? A lot of the way that tenure decisions have been made in the past is you're either first author or your last author, or it doesn't really matter what happens in the middle. In addition, some investigators who have been doing this for decades, a lot of their scientific clout and reputation comes from the fact that they are experts in the field, that they have accumulated hundreds of samples and have reputations and with the families and that's sort of who they are. If they let that go, their sort of clout in the community becomes different. Other barriers are more technical. So many of our samples are collected in family meetings. So the way that these family meetings work is there's a national meeting of the Williams Syndrome Association where every 2 years families from across the country and actually across the world come to a particular city. There are scientific discussions for the families about what's changed in terms of the health care for their children. And the families use what is basically their summer vacation to come and learn more about the syndrome, but also to graciously donate part of their time to research or to our research protocols. So research is conducted often in hotel rooms, and so we tend to try to use the least invasive methods to collect DNA on somebody's summer vacation, which often includes cheek swabs. So as anyone who has worked with this material knows, you don't get gobs of DNA. You also do not get renewable sources of DNA. And so the samples are limiting in terms of what people are able to share and ship out to somebody else. Again, as we've talked about it, often journals are not interested in publishing small cohorts of rare diseases, and we're often, even with using these group situations, not able to obtain the number of samples needed to put together a robust statistically group of samples. The other problem with using these family group situations is that often you will have 6, 7, 10 research groups conducting research at the same meeting, which means that the same 100 wonderful willing volunteers are having very dry mouths, giving DNA to multiple groups. So even if you and I decide to get together and collaborate our data and put it together, it is often not uncommon that we have many of the same people together. You don't want to find out that your person number 137 and my person 276 are the same person after you've spent thousands of dollars exome sequencing their data or their DNA. So what can be done? I think that there are a lot of opportunities for things to be done. I think a lot of it is going to involve changes. Like I said, most of the researchers talked about regulatory sorts of things. So can IRBs be changed in a way to allow dynamic conversation to occur between families? So like I said, the Williams Syndrome Association uses a registry, which is an online form where there are the possibility of dialogues where an email can be sent to families saying, there are changes to the protocol. Can you take a look at it? Do you want to see if you want to continue on with this? Now, having worked in collaborative research where we have had to use IRBs at multiple institutions together, that is an incredible challenge. What's okay at one institution may not be okay at another. And certainly with these newer technologies, that becomes a bigger challenge. But I do think using social media and using these things for families and the researchers to continue to talk over time only leads to better acquisition of data over time. Again, we talked about what happens when you actually finally get enough people to come together to put their samples together to come up with an exciting publication is that you get your 1,000 medulloblastoma samples and then you get an author list that is so big it doesn't fit on the paper. What is your academic institution going to do with your being author number 77 on a list of 173 in terms of what that work means for your academic progress? And so there need to be changes, and there are changes going on at promotion meetings, institutions at this time, but it's sort of just a new set of rules that are going to have to happen in academia itself. Funding organizations. It is my opinion that funding organizations should work to expand biobanks and those sorts of things for rare diseases. They are things that can be spearheaded by individual rare disease groups, but many rare disease groups do not have the financial structure to put something together. In the end, I think that because of this repetition of the same data over and over again, ultimately science will be pushed forward by having that all of that data in one place, but it's going to require infrastructure to do that. I also think that when samples are limiting, we need to to consider not just biobanks that can send out samples, but potentially doing all of the genomics up front, having the data then available that can then go back and be mined. In these sorts of situations, people will have to do their own phenotyping themselves, but still being able to go directly to the genomic data, potentially using unique identifiers that can be identified by multiple groups and then studied is going to be the way to go in rare diseases. And then I think a lot of it comes back to what families can do. I mean, in general, this is our careers that are taking place, but in general, what we're doing is for the benefit of the families that we care for. And so what can families do? I think what family groups can educate their members on the pros and cons of data sharing. I think in general, individuals who have rare diseases are willing to give up a little bit more in terms of privacy than potentially individuals with hypertension or other things because of the complexity of their disease, because of the severity of their disease and what finding a treatment means to them. We should talk to them about having individual members talk to their researchers about, are you sharing? All of us have seen a consent document that is 15 pages long. No one reads it. They sign it at the end. But at least talking to individuals about talking to the researchers. So are you going to share my data? Who else is going to be able to look at this so that more can be learned from it? At least if there is that discussion going on and researchers are aware that the individuals giving their time to the study want the data shared, may put more impetus on the researchers to make it happen. Again, creating databases with unique identifiers will be helpful. Journals should continue to require open access or accessible data, and it may also be possible to connect individuals with underpowered research so that data can finally get into it, versus allowing publication of data that maybe isn't fully powered so that individual groups can put their data together. Again, it doesn't serve our rare disease community or science itself for all of this data to be sitting in people's drawers. So in summary, I think the development of personalized medicine strategies for rare diseases is going to require large data sets. That's going to require coordinated efforts in multiple groups, and there's likely going to need to be changes on all sides in order to make this happen. Thank you.
Thank you very much. Our final speaker before we have the panel is Josephine Briggs, who will be providing a federal research funded perspective from the National Center for Complementary and Alternative Medicine.
Well, this superb workshop has been filled with important advice for federal funders. The issues that are being talked about here yesterday and today are very central in the eye of NIH leadership. We had a leadership meeting on Thursday and Friday, and a day essentially was spent on issues related to big data and data sharing. And there are just so many moving pieces in the things that need to be done to promote data sharing that I think my comments are only, in fact, going to cover a few of the highlights. Dr. Kozel's story of the challenges she has personally faced illustrate a number of them. Many of these do have partial fixes in the works, but there is, in fact, incredible need, as has been talked about so effectively in the last day and a half. Could I have, or here, I guess I do this. So as I think all of you are aware, NIH has a data sharing policy and implementation guidance. It's now almost 10 years old, and it has certainly been a building block in the issues that are being talked about today. A separate public venture for data sharing under different legislative authority is clinicaltrials.gov. And the authorities for these two activities are not identical and have some important differences. But certainly a point that's been made here today is the incredible impact that journal policies had in increasing the impact of clinicaltrials.gov. So we, in all of our areas where the funders can make a difference, partnerships are going to be absolutely critical. And our legislative authorities to do things, sizable as they are only effective when they are amplified by a variety of kinds of partnerships. In many ways, I view the data sharing policy as a very modest baby step in the things that need to be done in this arena. And I think most of my colleagues at the NIH, other IC directors, would share that perspective. I am also temporarily leading the CTSA program. The CTSAs are another setting in which we have and continue to make major investment in data sharing. Part of data sharing is making sure that academic institutions are taking good care of data. Certainly a major problem for the whole data sharing movement would be data that is not properly shared. Elaine Collier, who's here with me today, has been an important advocate for the need for policies in data sharing that ensure uniform and effective implementation of appropriate protection of data sets. And this is an area that we are thinking and worrying about, particularly in the setting of our investment in academic medical centers through the CTSA program. Another part of our investment in data sharing is the creation of electronic tools for data sharing. Some of these have been mentioned in the context of the last couple of days, last day and a half, including I2B2, which was discussed by the Johnson & Johnson representative as a useful tool for consolidating data sets, IDASH, which was discussed this morning, REDCap. But I chose in pulling together some information for this talk to mention three areas where we are investing in data sharing. And just to talk very quickly in the 10 minutes I've been allocated about what is happening in these three arenas. First, we are investing in data standards. We had lots of discussion yesterday about the complexity of development of data standards, and it is indeed a complex and difficult process, as many people in this room are well aware. The approach of getting a bunch of experts and locking them in a room and hoping that data standards will emerge is one that's widely used and kind of works. You get expert-driven medicine out of it. Whether or not an effective consensus comes together and whether or not one ends up with competing sets of data standards is probably partly dependent upon the ability of the funders to provide some leadership and direction. And some parts of the NIH have been pretty effective at this. Some communities have been better at this than others. But certainly an important part of data standard development is, again, a collaborative activity with the communities of experts, and it is in fact communities. There is usually not a single group who cares about the data standards. This is one example of a common data element development process led by NINDS. Walter Korschutz mentioned this yesterday. It's, I think, been quite terrific. NINDS is now mandating utilization of data standards in those disease areas where they have successfully developed them. On the other hand, there are ways in which the data element sets are specific to disease communities, and sometimes there's not agreement on how you describe demographic variables in different data sets. So we clearly need a continued push in the area of data standards. And my colleagues in the National Library of Medicine lead an activity called BMIC, which is a trans-NIH group that is collating currently data standards, at least to produce a list of the Tower of Babel, of all the languages that are being spoken. Another type of NIH investment in this area is actually creating data sharing resources. And again, I think this is an important area where those of us with leadership jobs at the NIH realize much more could be done. Here are a couple examples. These data sharing resources are largely disease specific, led by one or other institute and center. The NIDA has been the National Institute of Drug Abuse has been very active in this area. The sharing of data that come from clinical trials in drug abuse is highly sensitive. It's an area where both investigators and patients have been very cautious about sharing, but the development of data sharing approaches and an expectation that major studies share data has been very effectively implemented. This illustrates I think the complexities of NIH policy in the area. The data sharing policy calls for a data sharing and monitoring, a data sharing plan for every study that has a budget over $500,000. The other area that NIH is able to essentially require data sharing plans is in cooperative agreements, and many large trials that are run by the NIH are funded as cooperative agreements, and in the beginning stages of a cooperative agreement, it is now increasingly customary for the study group to develop data sharing policies, and then those essentially become mandates. In investigator-initiated studies, particularly those with budgets under $500,000, our policies are not as clear as to what authority we have to set expectations for data sharing. And clearly the strongest incentive we have is that we do control the purse strings. And by setting expectations, even in smaller grants, that data sets be shared, this is a strong place to stand. Nonetheless, in many institutes, there is some reluctance to utilize data sharing expectations as part of our mandate. A place where that reluctance does not exist, and one that I have considered to be a real model in oversight in this area, is in Heart, Lung, and Blood, which has for a long time set very clear, unambiguous expectations for data sharing in large trials and to a certain extent even in smaller trials. And they do fund a large coordinating center with a capacity to solve a lot of the problems that we've heard discussed in the last 2 days about data sharing, including review for what the IRB consent is, appropriate to sharing the data, including the preparation of de-identified data sets. I think the experience in Heart, Lung, and Blood is that by and large, in spite of this big investment in resources for data sharing, that a lot of the most effective data sharing occurs as collaborations, occurs as a dyad or group efforts in which the original investigators are part of the continued use of the data. But certainly many useful analyses are occurring with the BioLink as a very valuable starting point. I mentioned this is a committee, a trans-NIH committee. Dr. Collier serves on this committee. It is led by the National Library of Medicine, and they are a working group to improve the implementation of existing policy, to develop a consensus on best practices for program staff, to develop a guidance document for awardees on data sharing plans, to catalog the common data elements, and to encourage alignment between the standards being used in places like SNOMED and LOINC, and have those align with the data sets that are being used for research. Huge challenges. A new initiative that's being discussed extensively is one you've heard about in several settings in the last day and a half, a notion that there should be an ID for data sets. Dr. Goodman mentioned this idea. It has also occurred. Mike Huerta at the National Library of Medicine has written a position piece on this. And the notion that we should move to data sets that are put into shareable formats, carrying with them an ID and the potential for individual credit, I think is a very good and provocative idea. One last piece of the things that we do in this arena, we do provide a variety of solicitations that fund people to do secondary analyses of existing data sets. These are just a few examples pulled quickly from the guide, but certainly an important area. I do think that I just picked up today's Science, and I read from Bruce Alpers an editorial about the importance of small science, a very powerful piece talking primarily about small science in basic biological research. But the secondary analysis of data sets is an incredibly important way for individual investigators to participate in the generation of new knowledge about clinical subjects. And I do think that the kind of data sharing that we're talking about is absolutely critical for young investigators, indeed for all investigators, in improving the science of medicine. So there are many, many things that could go further than we have gone already, and this terrific meeting has certainly been very stimulating for me, and I will be carrying a number of the ideas back to my colleagues at the NIH. Thank you.
Thank you. Now we're going to move to panel discussion. If I could ask my colleagues who were previously up here to come up, I'll stand up so that there's sufficient chairs. And again, the microphone is open. Deb, before we get to you, let me ask one of the questions that came in from the internet viewing. I think we got enough room up there now. We do. So, Andrew, as you're getting settled, the question was first addressed to you, but I think that Steve Goodman and some others in the room might even have a comment. It said that Dr. Vickers, you noted that the authors of the majority of POLS papers where they agree to share as a requirement, they actually failed to share. So when you published your paper that pointed out that the journal was not compliant with its own policies, what happened? Was there any effort or discussion around the enforcement piece of it? And maybe we can get Dr. Goodman's perspective as a journal editor as well.
I guess the question is, did PLOS Medicine change?
Did they do anything about it?
Did they do anything about it? Well, other than trying not to publish our piece for quite some time. I don't believe so, no. And actually part of the problem I have with PLOS is that they felt it was a kind of, I was trying not to be antagonistic towards PLOS. This really wasn't about PLOS. This was really about the authors, and they felt very, very defensive about it. I don't believe anything has changed, and I think it illustrates a more general problem rather than anything specific that PLOS is doing wrong.
Steve, a discussion at the Annals of Internal Medicine, you said that you're afraid that you'll lose your authors. Is that sort of as far as the conversation goes?
Pretty much. We don't have any, and I don't know that I can speak for every conversation that the editors have had, but I'm pretty sure we have no recourse or enforcement for bad behavior, nor do we necessarily even have a way to find it out. I mean, that's actually what we were in the conversations I was having yesterday with the editors in anticipation of this discussing. But it's a very, very tricky area for a journal to be playing, sort of cop for behavior after the fact. And again, if journals were to play any role here, they'd all have to have some common set of behaviors and sanctions to do that.
Can I ask a quick related question? In policing clinicaltrials.gov registration before publication, has that been, do journals like do that?
Deb, do you want to answer that, Deb, or
Yes,
maybe help out?
Journals do that quite a bit, and in 2005, right after the policy was implemented, we watched and in fact at a DIA meeting, it went from 1 year where everyone was complaining about the policy to the next year. The buzz was, New England Journal and JAMA just rejected our manuscript and the whole room changed. But recently the estimate has been about a few hundred papers per year get rejected by the three U.S. journals that are in ICMJE, New England Journal, JAMA, and Annals of Internal Medicine. But the latest estimate was 9,000 that was published by somebody recently. So they do, and in fact, I get copies from at least Partners and Yale send memos out to their investigators every year that say, at Yale last year, I think it was 40-ish, 40 of our investigators were rejected, you guys really better register. And each rejection has a huge social impact, I would say.
Yeah, but I just want to underscore the fact that it takes all the journals working together and working together with you. No one journal could do this.
And Steve, it took some people, and it wasn't us for a change, but it was some other people outing the journals that were letting trials slip through. So in the early years, there were some people writing articles saying, X journal in the 12 main ICMJE members let their favorite trials slip through. So it takes constant vigilance.
Right, but I also want to point out that's actions that the journals have that's under their control. With respect to the data sharing, it's actions of the investigators that is completely independent of the journal control. So it's a bit of a problem.
So along that question, Miles, somebody specifically, the person who asked Andrew about the enforcement, they said, yesterday the Nature editor didn't mention this. Do you want to have an opportunity to comment on the data sharing enforcement piece?
Yeah, I mean, we can't go faster than the community, but we go a little faster than the community. So Nature Genetics, we have a checklist before we send a paper to review. We go through sources of data, whether there's a unique accession, whether it's live, whether it's available to the referees. That's very important. We let the authors know that this is a community requirement for certain areas, expression data, exome data, genome data, etc. Genome-wide association studies have been the exception, essentially because they're legacy cohorts which have been collected under particular IRBs. The individual level data have not been available to reviewers, have not been available, but there's been successful reuse by the consortia. So I think effectively what was just described by Dr. Briggs, that a lot of the data use arrangements are already in place in consortia through a sort of compete, collaborate, compel process. But then the next stage that we get to do enforcement, except in principle, we say your accession or your data sets are not available. Where's the data sharing plan? How are you making this data available? And then accept the paper. And then when it's up there, we get a lot of emails, and it's amazing how much data sharing happens afterwards. We couldn't get the data sharing plan, or it wasn't where we expected it. An email from the editor, and we got our data. The next sanction is either to have a data access moderation committee, as I mentioned yesterday, and if that fails, you can always put an editorial note on the paper, and nobody wants to see that on their paper because that's on the road to rejection, to retraction. So there are a number of sanctions. We don't want to use them. we want to use, the community, the current community norms, and those improve with time. We can't get too far ahead of our communities.
Thanks. Okay, Deb?
Hi, I had a couple of comments on Steve, and then Josie brought up one of these points. First, for the ID for the data set, I would offer a plea that if it's a clinical trial, use the NCT number, which is the unique identifier that you get in clinicaltrials.gov, which has currently a lot of utility. First of all, you can throw it into Google and you'll get everything, but it's indexed in Medline, and so it links through to everything. And it would be much better, rather than adding a new ID to the whole system, to use that. So I would like to make that plea. Second, in terms of protocols, let me just say a few things. Places like New England Journal have protocols for many of their articles. They're not, well, it still involves trust. They're not date stamped so that there's a date on them, but you don't know if they changed for example, the outcome measure the night before, or in Steve's anecdote, if the person who wrote the protocol the night before, made it so poof, obviously the outcome measures are going to be coordinated. One thing that will happen when proposed rulemaking is done, there will be time for public comment about the clinicaltrials.gov submission requirements. And one thing that HHS was directed to consider is whether or not submission of the protocol as a PDF would be required. And if so, when, at the beginning, at the end, both times, whatever. And that raises the question about what is a protocol. Industry has a lot of guidance about what has to be in a protocol. Non-industry has little to no guidance. And so those are areas where we'll need everyone in this room to comment. I don't know what the right answers are, but it'll be important to get comment from both the academic and the industry communities on that. So I just wanted to.
Yeah, certainly one of the key things that's emerged through the last day and a half is that, and that we've not talked a lot about, is what's going to be the education and training that goes into to this whole piece. Because the comments that, well, I didn't have a protocol or I'm still writing it, reflects a general lack of understanding of the clinical research process, and we as academics need to do a much better job about that.
And there's one area of great disagreement that somehow isn't brought out, which is when you need to have your statistical analysis plan. There are many opinion leaders, I'd say, who think it's okay to do it after the data are already collected but before they're unblinded, and others who just shudder at that thought. But there needs to be a discussion of that as well.
Yeah, discussion and training, and then there are public perception issues, as someone
Right.
pointed out yesterday, that industry-sponsored trials have a different view in the community, and yet the quality may actually be higher for some of the reasons that you've spoken about.
Yeah.
Thank you.
Just one comment on the ID. I think this is a very tricky area. The NCT is great, but the problem is some of the richest data sets come from ongoing cohort studies, which are not registered in clinicaltrials.gov, and we don't want to be in a situation where we have different formats for these IDs depending on what the design is or where it came from. In some of these cohort studies, they've embedded clinical trials, very complicated. So whatever we do, we don't want to reinvent that wheel, but we have to develop an ID that works for everybody.
So Sharon had asked us to sort of talk about the elephants in the room, and one of them seems to be this cavernous gap between well-intentioned policies and compliance with those policies in a number of areas, in the publishing area and the funding area. So there are some positive steps, I think, that could be considered. We've talked about challenges around academic advancement and sort of the models for that. One of the things that follows, in addition to publications, is funding. And so I think from the funder's perspective, you've provided a lot of good tools and policies about data sharing, which are being adopted, I think, in a variable way. Are there opportunities to sort of provide positives incentives, in other words, for people who are applying for new grants, considering track record in data sharing as a large chunk of the scoring in funding some of these proposals? That's one point. The second quick point, I had a question for Dr. Eichler. From the FDA perspective, the reason that we don't release subject level data is because we're prohibited from doing that by statute and regulation, and if we do, we get sued, and it has happened in the past. So that's sort of a legal barrier that we're left with. But Dr. Eichler, you had mentioned that you would be releasing full clinical data sets from both products that were approved and were not approved. It's the sort of the failed trials release that I had a question about. Are you running into individual country IP legal problems with that issue?
Okay, failed applications. I'm not talking failed trials. I can only talk about failed applications. So company X comes to us for a license application, we turn it down, or, which happens usually 5 minutes before 12, they would withdraw the application. Under European legislation, we are obliged to also publish that. So you will find an assessment report on a turned down application. Now, logic would extend that if that is in the public domain, and we say that clinical trials are not commercial confidential, that I see it happen, that there will be a request. Also, give us data on turned down applications. So it's not at the level of the trial, it's at the level of the marketing authorization application. Does that answer the question?
Yes. The other point related to that is how much resources you have to spend. I mean, from the FDA's perspective, even if we were able to share data and put it in a form, there's a process that has to be gone through to redact, trade secret information, patient, which is hugely labor intensive and really diverts from sort of the main focus of reviewing regulatory applications.
I can absolutely see what you're talking about. We are having the same problem. The current situation, what we call reactive. So you say to us, could we please have the data? And then that's reactive. We react to that by giving you the data. And that is a nightmare, because presently we also have to go through that. There isn't the idea is not about a commercial confidential, because in a clinical, we're talking clinical data here. We're not talking about the pharmaceutical thing. That's commercial confidential, but that was never been an issue. So clinical, we're not looking for commercial confidential. What we still have to do is the personal data. And as I say, we want to get rid of that. We want to make it a process that is not resource intensive for us, and therefore we want to be proactive. So it's on our website. You comply with condition A, B, and C. You click on it, you have it, and it should not involve any work for us. That's why we want to make sure that from day one, the data sets don't have any patient identifiable information in there. So that's the vision. When we will live up to that vision, well, future will tell.
We are coming over here, and then we'll go to you, Andrew.
Sorry. I just want to put that in perspective from the other side of the receiving end of EMA documents and some basic statistics and timelines. Again, the Reuters report that I showed you showed that they've released 1.5 million pages in 18 months. In our particular case of Tamiflu, it's 25,000 pages, unredacted, no redactions. So just to give you a sense of work involved, effort, and all that, I mean, some statistics put out there.
Andrew?
Yeah, on this point of personal data, my own view is that if there is no personal data, it was just the some ID number and their pain scores at the end of the trial, I think that should be totally fully open access. I don't think that we have to do the work to de-identify data, to take out personal information at all. I think that what we have to make sure is that if there is personal information, it only goes to certain individuals who have a duty of care. So if, for example, you're an academic and you have some question about the Tamiflu data or whatever, and you want that we can go through some process where we're saying that it's legitimate for somebody to look at this data, that they won't breach any confidentiality. Then I do not understand why we have to spend huge amounts of money deleting the names of individuals. This is a huge. I'm involved in it the whole time. I work with Swedish colleagues. In our collaboration, the Swedes can see that Niels Olsson died in 1991 of prostate cancer, and I can't, and I don't understand. We're part of the same research team. I have no personal interest in these men any more than my Swedish colleagues do. But because it's being transferred between different institutions, then there's this whole heavyweight regulation. In the NHS, when I was in England, and I don't know, there was this idea of a duty of care, which is within the national health system, you can get any personal information you like, right, as long as there's a good reason for it, and you have a duty of care not to abuse that. I don't see why we don't have the same situation for redaction and so on and so forth here.
Dr. Eichler?
Just a nuance on that. I fully agree. I mean, normally a clinical data set will not have the names in it anyway. Right. Why would you? No one needs that. The argument that is usually brought forward is you have a condition that is an ultra-rare orphan setting. There are just 2 patients with that disease in the whole of France. One is 75 years old, the other 25. Can you identify them? Yes, in theory you can. Right. Whether that is relevant in practice is a different matter. For a drug like Tamiflu, probably is not. So we'll probably have to address that, but there will be a way out. I don't see that as a stumbling block.
Hi, I'm Joe Ross from Yale. I just wanted to thank this great series of talks this morning. This question is directed to Dr. Briggs. I totally agree. The NHLBI BioLink website is fantastic. I've used it before. We've published papers off of it. It's been up for at least 3 years and very active, if not longer. That's how long I've been aware of it. What's slowing down the other NIH institutes from getting their big trials posted?
Well, I can't fully answer that. NIDDK has also established a data repository. It's actually older than that, although it doesn't have quite as strong electronic resources. In NCAM, we have provided funding to the major trials that we support, but they are relatively small in number, and we actually hopefully make the data sets available by paying the coordinating center to continue to put those data sets. And I happen to know that the GEMS and GATE and a couple of the other large trials are in fact available data sets. I think that there is a variety of things that leads to hesitation. One is a sense of all the hesitations, Dr. Vickers went through them very articulately, that the investigator communities have. So certainly one caution that NIH program staff feel is not moving ahead of the community opinion. But another, frankly, is resources. And the BioLink is expensive. And I was discussing it last week with Susan Churn, and she said, in fact, the most effective uses of these data sets are usually done in collaboration. A number of the Common Fund activities do make data sharing a very prominent part of the expectations. I'm also one of the leaders of a new initiative that was discussed yesterday that Rob Califf is heading to fund a set of pragmatic trials using healthcare system partnerships. And certainly in the solicitation for that, we made data sharing a very prominent component of the original application. So it's a culture change. And we have an NIH culture as well. And being very activist in setting expectations is something that's very much in the culture of some of the institutes and centers, and perhaps a little less so in others.
Terrific. Thanks.
Some of them are grants and some of them are contracts. Certainly setting data sharing expectations can be an appropriate part of a notice grant award, and so I don't actually view that as an important determinant of the difference.
Yes. My question is for Dr. Eichler. Assuming the EU goes forward and does in fact release a large corpus of data, in your estimation, will that in fact render about 90% of our discussion here today irrelevant? And I'm curious if you could comment on that.
Not 95, but probably 50%. Yeah, well, look, what are we talking about? We are only talking about clinical trial data or observational data in relation to a drug that is being submitted for a license application, or that is on the market and is undergoing relicensing, whatever. So anything that academia does, for example, in prostate cancer or surgery or so, is unaffected by that. So my first answer is, yeah, about 50%. My second is, will it set a standard? Probably yes.
Dr. Woodcock, did you want to grab a microphone and respond?
Ask Eric, though, generally you don't have the individual patient level data. So we're talking about release of summary data primarily.
Okay.
They don't have it.
Wait, we have it in a paper format. We will change that. We will get the full set.
I'll tell you, can I speak from my experience? They have it and they don't have it. We received that 8,545 pages was one of the 24 or so trials we requested. That one, they had it. The rest, they didn't.
So we're talking the future here. We will get it. We will request it from the industry. We will request it in a format that is individual, patient identifiable, sort of cleansed of that. And that will be it.
At the microphone.
Hi, Mike Watson from the American College of Medical Genetics and Genomics and a member of the Genomics Roundtable. I think that last comment highlights our problem is we pay for all this information in personal medical records, and we're unable to access it. And I'm not worried about the eMERGE study and the difficulties in that. There are actually other ways to get to the point of care and access clinical information. I direct NICHD's Newborn Screening Translational Research Network, where we have, as the coordinating center, we have our own sharing policies, but the individual grantees that operate in our system are obligated to write a sharing policy in their grant, as was mentioned. Too many of them say that we are going to give each other a password and share our data among our work group, and that's what gets through the review process. So I think much more robust kinds of sharing policies are going to be needed, really, to ensure access to the kind of data that at least publications have been based on. I want to expand a little bit on what Dr. Kozel said, which is she touched the very tip of an iceberg with Williams syndrome. We're talking 5,000 to 7,000 or so such diseases, and they don't all have a Williams syndrome support group that encourages the sharing of data and brings it together. So we have an enormous problem, and I think even if you found your 250 hypertensive patients out on the street, and then you went to look at their genetic etiologies, you'd find very few of each of the different forms of getting there and have the exact same problem. So I think we need to face the paradigm shift that genomics is bringing us and really look at how we access this data at a vast population level. Our clinical trials right now are to establish the clinical history of the disease, and then hopefully clinical interventions, not drug interventions. So I think we've got a long way to go to build a system that's actually going to work for where the genomics paradigm is taking us.
Dr. Kozel, do you want to comment?
I think I just agree with everything that you've said.
That's a relief.
Next at the microphone.
Yeah, hi, Jesse Berlin from Janssen. I have a question, but I was struck by a couple of comments in this discussion. One is with respect to protocols and what goes in a protocol. Steve, you may know that there's the SPIRIT checklist. There's been a working group that's taken a little long to come to fruition, but there's a publication coming out soon, just accepted for publication. The checklist will be in the Annals, and the explanation will be in the BMJ. SPIRIT is a group that was convened specifically for developing standards for what belongs in a clinical trial protocol. And it's detailed and explicit, and really speaks to content more than, does the statistical analysis plan go in the protocol or is it a separate document? So it's coming, and one of the purposes is exactly kind of educating people who are unfamiliar with kind of what is a protocol anyway, and the pro part actually being emphasized. And another point that Steve, you raised, since you mentioned observational studies. I mean a group of us in my department have actually published a commentary in pharmacoepidemiology and drug safety in response to a debate that's been raging in the epidemiology literature about the value or not of registration of observational studies. Where we come out is certainly in instances where you have an established cohort with pre-specified hypotheses and a protocol, where you're doing serious science, that ought to be registered the same way a clinical trial is registered and with results disclosed. So do you want to, if I can keep the microphone and after...
Well, Jesse, we got a long line and we got about 5 minutes left. So let Steve comment on that and then I'll go to Mike and we'll try to get people to...
I don't want to necessarily interrupt the flow. I'll just say one thing is that the important thing, I think, and this is a complicated issue, is that the data set be registered to have the ID and not a particular trial. So for example, from the MESA study, there might be 50 different studies and we could register each one of them, but the important thing is that it's the MESA data set that's being used, and that's a little different than the registration of the studies themselves. So I think that we have to find some way to talk about that.
Mike?
Mike Rosenblatt from Merck. My question is for Dr. Briggs. You spoke about the NIH thinking about data sharing, and I think that was very helpful for us to hear. But it occurs to me that one of the themes that we've been hearing about is before the sharing of the data, the problems of irreproducible data and how big a problem that is. And I'm just wondering, before we share data that may lead to misinformation or mislearning, that as the NIH is the sponsor of so much of this research, you must have concerns about the growing drumbeat and publications about irreproducible data. So could you tell us, are you thinking about that problem as well and what you're thinking about it?
Well, it has awakened enormous concern, and at least 3 groups have put together meetings talking about this. So there's a meeting of leadership from the Cancer Institute. Last week, there's a group dealing with this issue in NIMH and mental science, mental health. And so and Joe Ross's, who is here, paper was recently discussed in great detail at the leadership forum. There are many pieces of this problem. There's the problem that was talked about a lot last week was also the failures and difficulty in reproducibility of preclinical data. What we're talking about here together in the last day and a half has largely to do with clinical data, and I think that and perhaps to stand back, some of the spur for data sharing came from a very simple kind of data, namely genomic data. Now, genomic data comes with 4 letters, right? And we actually know a lot about the error rate in sequencing data. When we talk about the more complex types of data that we're talking about here today, the quality of data becomes a much bigger problem. How do you even measure the quality of data? I'm a believer in transparency, and I think what's being talked about here is only part of the fix for the problems of reproducibility. But certainly the NIH leadership has been paying intense attention to these issues, and it's been a subject of enormous concern to all of us.
So let's go with, and I'm going to apologize, one more question, and then we do have another 15-minute question-answer period that we can come back to, and I think most of these folks will still be here. So let's go one more.
Hi, Michael Cantor from Pfizer. This is for Dr. Goodman. When you were showing the policies of the other journals, you noted that Science, Nature, and PNAS have all those policies in place and they're actually able to enforce people to submit their data, whereas the medical journals don't. And you noted that you can't really enforce that because people will submit to other journals. So how do you learn from the earth sciences or the hard sciences over to the clinical journals to kind of have a parallel, I would say a set of rules so that you can increase participation and get people to submit better? Is there something different between the two fields?
So I'm not a sociologist, and they are quite different for many of the reasons that have been brought up. I would say that it's absolutely critical within, and we heard a lot about the community, and within the genomics community, obviously they what they give up, they also get back, and that hasn't been, and that was actually the theme of the meeting of funders, that they have to develop a culture where the sense is that even though you're giving up something, you're getting back just as much, and that you will have access to these collected data sets as well. I think it's a little bit too complex for me to, I can't, it has to do with academic incentives, it has to do with regulatory, it has to do with a lot of things that have to come together to force collective action. It has to be both push and pull. It can't just be forcing people to do something. I do think that it has to do with the community understanding that it has a lot to get out of it other than just punishment if they don't follow the rules. And I think a lot of comments here have talked about ways to get there, but it's going to take all the interested forces to be working collectively, developing metrics for, I love the comment about the NIH giving credit for data sharing. That would be part of the solution, promotion, part of the solution, and many of the things that have been said here, but I don't want to cut short Janet's.
Janet,
please.
It has to be very quick.
We have 159 million lives in our Sentinel network now. So people are talking about observational studies. We're definitely willing. We've been going out begging people to figure out how to share this national resource to allow other studies to be done on this network. So that is a model that is for the existing healthcare data that is highly valuable and has a huge number of, but everybody wants to do their own thing and nobody wants to join together. It's probably what you've been talking about all morning.
And we can come back when you join the panel at noon to further that discussion. Well, let me thank the panel and take a 15-minute break, and we'll come back and get started.